this video, we will study about the chromosomal basis of sex determination. The first clue about the role of chromosomes in sex determination came from the experiments which were performed by Henking in 1891 during his study of spermatogenesis. And during the process of the study of the spermatogenesis, he observed the movement of certain object, and he found that only 50% of the sperms received this, while the other 50% did not receive that. So he called that body as X body, which was later named as chromosomes or X chromosome. The X chromosome is also called as sex chromosome because it decides the sex of a progeny. Apart from that, the rest of the chromosomes are known as autosomes. So, the movement of this enabled us to form the chromosome as the basis of the sex determination, the type of chromosome. Let us study the few types. The first type which we are going to study is XX and XY type. This type is seen in the case of human beings. Here the female is having 2A plus X and X. So these are autosomes, and these X and X, which are the sex chromosomes, a female is there, where a male is X and Y. Sorry, this is male. During the formation of the gametes, the female will form X and X gamete, whereas the male will form X and Y, and during their crossing over, it will result into progeny which will be 50% male and 50% it will be a female. So here these two will form a female, and this will be male. So the chances are 50% male and 50% female. This is seen, XX and XY type is seen in the case of human beings. In the case of birds and butterflies, we find ZZ and ZW type. We are not using XX over here so as to avoid the confusion. Now, if the chromosome set, that is the sex chromosome, are same as in the case of this, as in the case of this, then it is known as a homogamy condition. Homogamy means over here the female is showing homogamy, whereas the male is showing heterogamy. Here Z and Z, that is a male over here, and if the two chromosomes, our sex chromosomes, are different, it is a female. It is seen in birds and butterflies. So over here the male is showing homogamy, whereas the female is showing heterogamy, and in the case of human beings, the female shows homogamy and the male shows heterogamy. Now here Z and Z and Z and W, this is a situation where the homogamy is seen in males and heterogamy is seen in the females. The third type, which is X and X and XO type. If it is a homogamy condition. It is a female, and if there are no other X chromosome, then it will be a male. Such a condition is seen in grasshopper, where X is not being transferred over here, and as a result it will have only one X chromosome, resulting into the formation of male. Another type is seen as another type is haplodiploid type. This type of sex determination is seen in the case of honeybees. Now here the female is having X and X over here, and what we say is if the female has two sets, then it will be a like 2n plus 32, and the male will have n plus 16. Now on the gamete formation, this will be a female and this will be a male. Now on the gamete formation, 16 and 16 over here, and here only 16. So if the fertilization takes place normally, it will result into the formation of zygote, resulting into 32 number, and this will develop as a female. Now here this is undergoing, this is not fusing with other gamete, and so during the process of parthenogenesis, that means development of individual without fertilization, it will result into a progeny having 16 chromosome, and as a result this will develop as a male. So in the case of honeybees, the males are developed if no fertilization takes place and only through the process of parthenogenesis, whereas a female is formed or is obtained when the fertilization takes place after the gametogenesis, and the zygote formation results into the development of a female individual. So there are 4 different types of the sex determination which we have studied, that is XX and XY type. The example is human beings and Drosophila. ZN and ZW type, it is seen in birds and butterflies. Then we have XX and XO type, where only one is being transferred, one of the X chromosome is being transferred, and this results in X0, which is not having the X chromosome or not showing homogamity, will be a male. Haplodiploid type is seen in the honeybees, where the males are developed after parthenogenesis, that means they are developed without fertilization, whereas the females are developed after the fertilization. So hen king played an important role in our understanding of the process of sex determination.